introduce to you the mini-tube AndroVision eFlow system today.
The reusable eFlow chamber is the heart of the eFlow system. It is a precision-engineered piece of equipment with a small space and viewing glass for the semen sample inside. The chamber is mounted on the microscope stage but can be removed easily any time. The actual space for the sample is divided in two, providing a small and a big gap for the sample. The two gaps produce a thicker and a thinner layer of semen. The system measures motility and concentration of sperm in the thicker layer, capturing a great amount of sperm in every field to provide an exact result within short time. The thinner layer is for analysis of morphology defects with the AndroVision automorph system, which detects the amount of distal and proximal droplets and of bent tails in the sample. The thinner layer makes this analysis very precise. This is the eFlow tower. It is the brain of the system. The tower controls the flow of liquid into the chamber and out of the chamber. It has a diluent reservoir included, which keeps the extender warm. The mechanism in the front of the eFlow tower aspirates the correct amount of the semen sample from the sample container and makes it flow into the chamber. There is no need for a pipette to load the chamber. After each analysis is finished, the eFlow tower empties the chamber and flushes it with extender.
At first, I would like to show you all the components that belong to the eFlow system. First, the heated extender bed with the control unit and the dispenser. Then the control unit for the warming plate and the microscope stage with the stands for the eFlow containers, the electronic mixing pipette, the eFlow tower and the eFlow chamber, the microscope with the automatic scan stage, the USB high-speed camera and the CASA system AndroVision. Now let's start with an ejaculate analysis. The ejaculate enters the lab. First, it's important that you mix the ejaculate very well. You take a sample container and pre-fill it with heated extender. Take the electronic pipette and take the semen sample. It is important to wipe the tip and then mix the semen with the extender in the cup. So, you select the bore, take the eFlow sample container and put it into the eFlow tower and press OK, and the filling of the chamber starts automatically. Now you have to check the focus and the light and press Analyze. The system automatically analyzes 4 analysis fields for automorphology and another 3 analysis fields for motility and concentration.
Here in the thinner layer of the chamber we have a very detailed and good picture where the system detects proximal and distal plasma droplets and bent tails, and they are marked with these colored boxes. On another 3 analysis fields, AndroVision records motility and concentration. Therefore, the system analyzes a higher number of sperm cells in the thicker gap of the eFlow chamber to get a very precise result for motility and concentration.
After the 7 analysis fields, the eFlow tower starts automatically to flush the chamber and prepare it for the next ejaculate. During the flushing process, eFlow chamber, the user can check the results and then accept the data to come back to the donor selection list. For quality control of your produced semen doses, you can prepare the samples directly out of the semen tube. Therefore, you mix the tube very well. Open it. And now you fill the eFlow sample container approximately until the first mark directly out of the undiluted semen dose. And put it into the stand on the warming plate to pre-warm it for about 20 minutes. After the 20 minutes pre-warming time, you take the sample container out of the stand, close it with a silicone plug and mix it carefully overhead before you put it into the eFlow tower.
With the quality control module in AndroVision, you can check the quality of your produced semen doses with the eFlow system. Therefore, you select the date of production of your doses you want to do the quality control. Now you get a list of only produced bores on the selected date. Then you select the bore and you see here the ejaculate line. Now with the eFlow you have two options. You can either analyze motility concentration and automorphology like in the production mode, or to reduce analysis time you have the possibility to set the analysis fields for automorphology to 0. That means you just check motility and concentration for your quality control. The eFlow chamber is filled. You focus the semen and check the light intensity and press Analyze to start the measurement. Now 3 analysis fields for motility and concentration are recorded. On the right side of the screen, you get your results, always live during the measurement, per single analysis field and for the complete sample. What we see now is the result for that quality control. We analyzed 860 sperm cells. We have a concentration of 0.022 billion per ml. We have a total motility of 98% and we have a progressive motility of 82%. To save the data, you click on Accept and you come back to your filtered bore and ejaculate list of the 28th of April to run the next measurement. The AndroVision standard reporting provides you several options to export analysis or production data and create certificates for single ejaculates. For example, you have daily production lists available for export either to Excel or to create overview reports filtered, for example, by species and breed. Another possibility is to create certificates of single measurements, single ejaculates, where you have different possibilities to decide which information you want to have on that certificate. You can design your own headline with the lab information and the logo. You have the animal information on the second part of the report, You have an analysis picture, you have the analysis data, you can decide if you want to have the concentration as a result on the report or not. So you have flexible possibilities here in the creation of these analysis certificates before you save them as PDF files. For quality control samples, you have the possibility to create analysis certificates with the results of the quality control measurement with AndroVision. You see the date of analysis, you see the date of production, and you can create a comparison report when you select here the option Show Native Values. The system creates a report where you have both results. That means the result of the native ejaculate measurement and the quality control result next to each other to compare these two results.
The AndroVision eFlow system is robust, precise and simple and stands for automation and standardization in semen production.
Hi, Sam. I'm the lab manager for the Borsteds at Triout Foods. Since getting the eFlow system, we have noticed an increase in the accuracy of our concentrations compared to the nucleocounter and third-party audits. The training has become easier and much quicker. We're happy with the incorporation of the eFlow system.